Hi, I'm Max. Today we are going to be talking about gel electrophoresis, which is a process which is essential for any genetic laboratory. The goal of gel electrophoresis is very simply to separate macromolecules such as DNA according to their size. It measures the strands of DNA or an RNA in terms of number of base pairs. When we measure the length of a road, for example, we use a unit such as kilometers or meters. Base pairs is the unit that we use in genetics and in biotechnology to measure the size of strands of DNA, which are very tiny things. This process of gel electrophoresis separates molecules such as segments in DNA, and because it is important to know the size of DNA and size of small molecules, it is a fundamental for any genetic laboratory. There are a few important things to note when we're talking about gel electrophoresis. First of all, look at the setup. There are a few important things. We have the agarose gel, the power supply, and the electrodes, which are basically the essential things and the way that this thing works. The agarose gel is the medium in which the process of gel electrophoresis occurs. If you look at the image on your left, there is a blue square with holes inside, which are the sample wells. This is the medium in which the process of gel electrophoresis works. It is a porous substance. Very simply, it is similar to a solid structure with funnels. We will get more into this later on in this presentation. The power supply sends off positive and negative charges to opposite ends of the agarose gel. It is the box in the top right corner. It has wires coming out of it which are black and red. And finally, the electrodes are the location where the charge is sent. It is located on the electrophoresis tank. One electrode, the positive one, is red. The negative electrode is black. A charge is sent from the power supply to the electrodes, and this is the process and the way in which the whole thing of gel electrophoresis works. Very simply, DNA strands have a backbone of phosphate, which carry negatively charged oxygen molecules. Therefore, the overall charge that occurs on a strand of DNA is going to be negative. We know that negatives repel negatives, and positive attracts negatives. If we go back to our diagram from the previous slide, one side of the buffer solution is given a positive charge, the other side is given a negative charge. The wells are on the side of the negative charge. DNA, when the power source is turned on, the charges are sent to each electrode, and DNA is repelled from the negative end and attracted to the positive end. The process of gel electrophoresis uses an electrical current to separate DNA molecules. So these DNA molecules travel through the electrophoresis gel. You probably recall of when at the beginning of this video we talked about how agarose gel is a porous substance, meaning that it has small tunnels or funnels that exist inside. Think of it as a net. Larger molecules have more difficulty compared to smaller molecules to travel through the gel. It is like a funnel. The smaller the molecule, the larger the distance that it can travel in a given time. There's less resistance. Therefore, the larger molecules, meaning the ones with more base pairs, end up closer to the negatively charged end compared to the smaller molecules which end up closer to the positively charged end. The molecules are repelled and they are sent through the gel electrophoresis because they are attracted to the positive end and repelled from the negative end. There are the funnels in which they have to pass through, and if they're smaller they can quickly go and it is way easier, there's less resistance. Therefore, at the end of this process, the smaller the molecule, the further it will have traveled through the gel. At the end, the size of DNA strands can be taken according to the number of base pairs. This process is quantifiable, and we can figure out exactly the size of base pairs of the DNA molecules. Once the smallest fragments of DNA have finished migrating through the gel, the process is complete. Therefore, our power source is going to be turned off. We turn off the power source. The larger molecules of DNA are going to be closer to the starting point, which is at the negative end. The smaller molecules are going to be closer to the positive end, which is the far point. They have traveled further in the dye during the process. Because we cannot see the results of gel electrophoresis, we add a DNA binding dye, which when it binds to DNA, it glows in UV light. We put this under UV light so that we are able to see the results with our own eyes. data shows up as bands, each containing thousands of identical DNA molecules of that length. So when we finish, we add a UV light to be able to see the results, and we can track our results and determine the length and size of the DNA fragments. Gel electrophoresis isn't just something that we do for fun. It also has several practical uses and is used in the scientific field. It is used for anything that requires or could benefit from the separation and classification of DNA molecules according to their size. It is used in things such as DNA analysis to investigate crime scenes, the analysis of genes associated with particular illnesses, any rudimentary genetic analysis. So when they have something with genes, the first thing is to do PCR for the rudimentary genetic analysis of the gene. And these days, it is currently being used to create dichotomous keys and classify organisms according to their genetics. The technology of gel electrophoresis is also used to analyze the contents of plasmids using restriction enzymes. And it is also used to analyze the results of PCR, which stands for polymerase chain reaction. I'm not going to go in depth, but it is just amplifying a wanted gene using thermal cycling. Gel electrophoresis is also used in DNA and protein analysis, and it could also be used in the real world to find the molecular weight of viruses and partially or fully map them, as seen in several new studies. Gel electrophoresis has been one of the most useful technologies in scientific research since its adoption in 1970, due to its extremely versatile technology. It can arrange proteins or macromolecules based on their size and number of base pairs. It is essential in any biological laboratory because it does the first things that most scientific research does, which is figure out what they are dealing with and the size and what it is. Hi, thank you very much for watching all the way till the end. Here's a list of the works cited that I used for this project. I did a lot of external research, and this is a list of sources that I used, publications and studies. I'm Max. Thank you for watching. I hope you have a great day.